afternoon, everyone. Welcome back here at the ESCMID Global TV studio. My name is Judith Cohen. I am your host here at ESCMID Global TV, and I'm very excited to introduce to you my next guest, who is Professor Alessandra Carattoli, who works in general microbiology and the Department of Molecular Medicine at the University of Rome, La Sapienza in Italy, and who will tomorrow be talking about a very interesting topic on decoding plasmids in diagnostics and infection control. So let's just grab this opportunity and of having the great pleasure of you being here with us in the studio and explaining us and the viewers a little bit more about this interesting topic.
You want to know probably what is a plasmid?
What is a plasmid? I looked it up personally, but I can imagine that there are some viewers that do not immediately know.
Plasmids are extra chromosomal molecules. That means they are not in the chromosome, and they are outside the chromosome in the cytoplasm of many different bacterial strains. They replicate autonomously. They are able to promote their propagation by conjugation. So they transfer from a bacterial cell to another bacterial cell. For many years, they have been a good research field for studying DNA replication, the conjugation. So they were basic and on the academic benches, mostly.
Okay. Because what is their main role with bacteria? So they make sure that the bacteria can
It's sort of
survive?
symbiont parasite, because they promote the diversity, because they move DNA from one cell to another. But currently, they are changing completely their role, because they are now a public health issue. Why? Because they have acquired antimicrobial resistant genes.
The plasmid.
Yes.
So this is an extra chromosomal genomic piece of information.
And transferable, not only present in a cell, but these antimicrobial resistant genes located on plasmids are able to transfer. So they can eventually start in a non-pathogenic strain and then to go into a pathogenic strain. And also, this is the most important and clinically relevant mechanism for acquisition of genes that is for the bacterium cell is to win the possibility to survive if the antibiotic is used, and they have found a plasmid with the resistance to that antibiotic. So antibiotics are selecting for resistant bacteria, but also for plasmids carrying resistant genes. So they are now in a mutual situation for surviving, and they are evolving together also.
So how long are you working on plasmids already? Because maybe now they'll get more visibility, or people will be more aware of them since they've become a public health threat with regards to the AMR.
I studied those since 1997, more or less.
You're an early adapter.
I didn't count, but I was among the basic researchers, basic science researchers at the beginning. Then I was studying those with resistant genes, and I was one of the first to take care when they were not a problem. And you know, sometimes things happen, so they were evolving, enlarging, and becoming more and more relevant. So this basic science moved to in a wider interest for science and for clinical micro also.
Because you've done many studies on antibiotic-resistant bacteria and the plasmids that confer the resistance. But tomorrow you will be talking at 11 o'clock in Hall I for the people who have an interest here in plasmids. You will be talking about decoding plasmids in diagnostics and infection control. So it seems it's a little side step. Do you care to share a little bit more how plasmids are of importance with diagnostics also?
They are now part of bacterial typing. What does it mean, typing? Typing means that you want to know exactly the name, the surname, the content of a bacterial cell that is causing an infection.
Yeah.
And this is an epidemiological marker to follow these bacterial cells from a patient to another, from even at the wider global level, you can trace. Successful bacteria and also successful plasmids, because there are some plasmids that are epidemic, so you can find different kind of bacteria in different parts of the world, and they are very well conserved, like a sort of virus, but without the extra phase, so without an extracellular phase. And so there are a lot of methods that have been implemented in typing plasmids, and not only bacterial typing. So I elaborated some methods during my career to follow them. That's the reason why we say decoding and diagnostics, because some of these methods are coming more and more frequently into the practical life of a clinical
You developed kits to identify the plasmids
yes, it
also
was
in
originally,
certain bacteria.
Originally was a PCR, an amplification kit, basic kit. And then with the genomic area, it's evolving now in databases for identifying plasmids into the genomic sequencing data, and I think this is the progress that you have to do, starting from a molecular bench test to something that is wider and can be done everywhere. So,
So what will be the main focus of your presentation tomorrow, or your lecture?
The title they gave me and they proposed me is in fact the state of the art about plasmid typing. So I will report a bit of history of plasmid typing, starting from conjugation tests that have been done in 1970s. It was a phenotypic test, very boring, I should say, very long, very complex. Then the PCR methods, I did in 2005, and then we will move to genomic area. And in genomic area, there are so many important papers. I will bring also a review of current literatures on Nature, microbiology, published on March 2024, just yesterday. And, and I try to bring some overview of the state of the art of plasmid typing. And I think I will end with some examples of plasmid typing. This is
so you very well explained the relevance of acknowledging how important plasmids are with regard to a lot of different issues. For one, the least, or not the least, being the AMR. What challenges are there in this field of research? What are issues that you encounter?
The complexity, because the technology is evolving quickly, and there are currently more than 60,000 completed plasmid sequences to study. And
how many? 60,000?
60,000 plasmids.
60,000
other authors, Galata, Schwartz, created a database of plasmids, and where they collect all plasmids that have been fully sequenced. So we have these references. 60,000 means that you cannot study one. It's huge. And now the frontiers is to understand if there are some clusters, if they are evolving in a direction, in another direction, how do they change? Because this is also important, the evolution, because there were things 10 years ago that were not the same plasmids that we see today. And the combination on the same molecule, on the same plasmid, of virulence and resistance, this is happening also. And so it takes a lot of energy. Now it's becoming more and more a bioinformatic problem to analyze sequences.
Yeah.
So tools for these are daily published, and this is a limit because I am a biologist, a wet lab biologist, and now the young bioinformaticians are given lessons on how to pick up fishing plasmids within the genomes.
Wow.
Wow, yes. This is the they are doing this
job. It's good to see science progressing.
That's fantastic. Yeah. They do. I follow. Yeah. I follow. I admire, but I'm still on a wet lab.
Yeah, so tomorrow the session, it's being organized or co-organized by the Emerging Infections Task Force in ESCMID and the one of the study groups in ESCMID, I believe, the ESC-BOR, the Lyme borreliosis study group. So that'll be really interesting. Do you feel that this year's ESCMID Global, are there enough other experts from different countries also here to network with on plasmids, or?
A lot.
Is it a lot?
A lot. This is the genomics of bacterial pathogen is the emerging issue in this global ESCMID. At different aspects, I followed this morning Professor Holt, she had the keynote lecture this morning, and explaining how genomic is impacting the public health issue, and now we are rapidly, very quickly arriving to move genomics in the hands of medical doctor at the bed of the patient. This would be the frontiers to be very, very processive quickly. So it was an experiment. Now it's going to be a daily life. So every aspect of genomics.
It must be wonderful to see it progressing just from idea to
it's
end user.
It's now.
Actually,
it's happening now, and every ESCMID there are more and more sessions dedicated to databases, bioinformaticians, analysis, and so on. So I think this conference is particularly important for us because we meet colleagues personally, we exchange ideas and plan for funds together or ideas together, so consortia together. So this is 3 days of exchanges.
Very rich days. Well, thank you so much for taking the time and effort to join us here. For our viewers, if you want to spike up your knowledge or just or interact with Professor Carattoli more on this topic of plasmids in diagnostics and infection control, that's the name of the session, Decoding Plasmids in Diagnostics and Infection Control. It is tomorrow at 11 o'clock to 12 o'clock in Hall I. So make sure you don't miss out on that if that has your interest. We will be taking a short break here at ESCMID Global TV, and we will be back at 3 o'clock sharp, when we will be inviting another live guest here in the studio, who is Justina Kowalska, and she will be talking about the session that she will be speaking at on how you're never too old for an STD, for a sexually transmitted disease. So definitely tune in if that caught your interest. See you at 3.